So let's dive right in. Excuse me, I got a bit of a cold, so hopefully you can hear me. So let's see if I can move my slides forward. Okay, so I'm going to give you some just background here. So this is what I talked about, knowledge integration or curation. We'll start by saying what knowledge is. So here's some knowledge about a gene called fibronectin. You'll notice that there's a lot of it. There's 37,000 articles that come up in a PubMed search about this gene. We know, obviously, it's important for research to advance to be able to access knowledge like this, use it. And we'll notice that most of it, still to this day, is shared and represented as text. And you are going to want to use knowledge to answer questions like this. So what are the functions of fibronectin, which is a gene? And so you could go there and read through those articles, and you could, in principle, answer that question after quite a while. But what happens when you come up with your high throughput screen results in a list of 238 genes, none of which you've heard of before, and you have to answer the same kind of questions about them. All of a sudden, it becomes almost impossible to do it that way. And so what you obviously need is a way to translate that text into answers to that question. And so that these things, can you see my mouse? Does that come through?
Yeah.
Hope that it does. Yes, okay, good. So these things that look like that are called knowledge bases, or in some terrible situations, Excel tables. But anyway, in the process of building them, I'll call knowledge integration or curation or biocuration. Okay, so there are lots of knowledge bases in the world. Everyone that is in science has been exposed to some of them at one level or another. Probably the most famous of all of them is the gene ontology, and so I'll spend a little bit of time talking about that before getting into the Wikidata stuff. And so why is it important? So these things are important because of how they're used. So applications of knowledge bases include, obviously, finding information. So finding information about the function of a gene, for example. Another perhaps underutilized aspect of knowledge bases is for planning research. So what genes do we not know the function of? Maybe those would be good research targets. But the big one here is interpreting data, especially in the world of high throughput data generation. And so this is kind of the focus, and really gene ontology enrichment analysis is probably the most commonly applied use of knowledge bases in the context of biology. And so the way this basically works is that you have some interesting list of things, for example, genes. You apply some, I'm not going to go into detail on this, but you apply some interesting statistical algorithms that take advantage of the gene ontology knowledge or biological pathways or other kinds of knowledge bases, but ultimately which are ultimately very much the same kinds of ideas. And that turns into one of those pretty pictures you see on the right that gives you a very rapid way to understand, at least at the surface level, what's going on in the experiment that produced that list of genes. And this has become a very important part of how we do biology now. I think as evidence for that, you can look at the number of citations of the gene ontology paper, which is now approaching 16,000 and grows by more than 1,000 a year now. Most of those citations are related to its use in high throughput analysis of data at this point. And these problems will only increase in importance over time, clearly, which is fantastic, right? But of course, there are problems, right? So number one problem, which I'll go and give you an example of, is that knowledge bases are not complete, and I'll use an example from the gene ontology to illustrate that. So here's a gene called the 5-HT6 receptor. Okay, in case you didn't know, that means 5-HT receptor means serotonin receptor. Okay, here is an entry about this gene coming from the Gene Ontology Consortium. You'll notice there is receptor activity, but there's nothing to say that this is related to serotonin receptor activity here in the annotation. That's obviously a big problem, right? And I'll point out that, so this was actually from a slide I generated for a different talk in 2010. I went and looked this up again, and now, as of 2016, they did add it. So it wasn't that it was somehow incorrect. It was just that it was missing. And that was added in January of this year. And if you look back in the literature, that gene and its connection as a serotonin receptor was first characterized in 1996. You can find that in PubMed, in that journal article there by Cohen in the Journal of Neurochemistry. Now, what this means when you come back to this, when you come back to your use of this kind of knowledge, these knowledge bases, is that all of these analyses are fundamentally flawed. Okay? There's a real problem here. So given your interesting gene list, your fancy statistics will give you these overrepresented categories of information about how the inflammatory response is activated, defense response is activated in that list. But wait a second, maybe you missed that serotonin receptor activity should have been there. And of course, there's so much more that you don't know. And so clearly, this is a big problem. And this leads to another problem, is that biologists, by and large, are really annoyed by the gene ontology. And every time you say it, isn't it great? They say things like, maybe it's not so great. But they have to use it because they don't really have many other options. And so, why is this the case? The case is the process that we have for building knowledge bases. So the process runs like this. Scientists do science, what they like to refer to themselves as real scientists when they talk to computer people. Then they publish it in modern publishing forums like this PDF here. And then somebody has the job of manually reading that paper and turning that into that knowledge base that we use and we depend on. So what's the problem? Why does this guy look not very happy? The problem is this. The problem is that we have vastly more scientists doing this kind of work for much greater rewards than we have data curators. I mean, I'm not making the numbers up. We're probably talking about hundreds of thousands of scientists that produce things that literally only hundreds of data curators need to process. And just because I can't do a talk without presenting this particular slide about the growth of knowledge, this is just to give you a scale of what they face. So as of last year, we were approaching 1.2 million articles added in PubMed. We passed 1 million articles a year just a couple of years ago, and you can see the direction this is going. So that's just to give you a scale of the amount of knowledge that needs to be processed and represented somehow. And so to sum this up, the introduction up here, professional biocuration does not scale up to the rate of production. Okay, not the way we're doing it right now. Before getting to Wikidata, there's one other problem I want to raise. The other thing, aside from being incomplete, knowledge bases are not integrated well with one another, and they need to be. And so if you are going to solve any particular problem in bioinformatics, you're going to end up taking information from one of these things and mapping it onto information from another one of these knowledge bases. So putting those all together is what we really want to do. We want to bring that together so that you have access to the sum total of biomedical knowledge as it stands today to do the best job in interpreting your data and generating your next hypothesis. The problem is that this is actually a really difficult problem. And one way of phrasing this is as the language barrier. So as one more example here, here is an item in an important database run by the National Library of Medicine called NDFRT, the National Drug Resource File. Okay. What it says is that something here called N0000000174 may treat opioid-related disorders, which is a useful bit of knowledge to have access to. Over here from PubChem, we have something with ID 409, has molecular weight and all kinds of other interesting chemical information. Over here we have a drug bank entry, DB0033, that tells you where the drug's manufactured, and again, all kinds of other medically relevant information. Over here we have PubMed, which has articles about something called methadone written in text and tells us what interacts with, what other drugs interacts with. All of these things, of course, are talking about the same thing, but it's very difficult to know that and be able to trust it enough to compute with it. And so that is obviously a problem. And so it's such a problem in bioinformatics that it's like closet industry. So if you're in bioinformatics, this is great. You know, it keeps people's paychecks coming in doing those sort of identifier mapping problems. I mean, if you look at, and you can even publish papers about it. If you do a Google Scholar search for bioinformatics and identifier mapping, you get 469 papers, which is obviously, you know, good for bioinformatics, I guess, but it's bad for science overall. So, you know, given that backdrop of where we are in terms of building knowledge bases for biomedical applications and supporting research, what can we do to make it better? And so, in this talk, I'm going to talk about the concept of using a global knowledge platform. The question here is, you know, this is a research project too, so we start with a question. What would happen if everyone was literally working on the same database? You think about it, just assume that we could be working on the same physical thing. If we took all of this knowledge and we put it into one pot. Yes, there are problems, but I think it would come with some benefits. One of the benefits would be that we could split up our work more effectively. At the moment, each of these different knowledge bases are, generally speaking, independently funded, don't necessarily talk to one another, and may often be doing overlapping work in a redundant way. That is not the most effective way to solve the problem. The other thing that would happen, I posit, is that we would make integration between the different concepts here the default behavior, something that happened up front when you're representing the data first, rather than downstream by someone else by accident in an ad hoc manner, which is quite different, which is the way it happens now. And this, of course, is what we're going to try and do with Wikidata. So I'm going to just take a second. So this is Wikidata. Wikidata is to data as Wikipedia is to text. It's a free and open repository of knowledge. Its central mandate, what they say when they could advertise it, is giving more people access to more knowledge. That's its goal. It's managed by the MediaWiki Foundation that operates Wikipedia. So this is not a product of Scripps or the SULAB or me. This is a product of the MediaWiki Foundation, and we are using it to do our work in biomedicine. So step one, it's a knowledge base. Okay, it's a knowledge base. It's ultimately what it is. It's a database. Okay, the difference about it versus other kinds of databases is that anyone can edit it and anyone can use its contents. All of the content within it is CC0, meaning that you can use it to do whatever you want, including launch your own business. There are no limitations on it. And so this is how the data is represented. So here's an example. It's represented in the form of statements. Okay, so we have within Wikidata, we'll have things that are called items, which represent some particular concept. So in this case, I have the concept of London, represented by its unique ID in their system, Q84. And what I'm doing is making a statement about its population. So within this statement, I have a property name. So there's a population. Then I have a value. I have 8.1 million, and that is my claim. And that claim comes with a context called a qualifier. So this is the preliminary estimate as of June 2012. All of that whole claim, including its qualifier, is backed by a source or a reference. Putting all that all together, we have a statement about London. So something to notice here is that you could have many statements about the population of London within the system. The system isn't necessarily about representing absolute truth. It's about representing claims about the world backed by evidence that you can go back and verify. That's its goal. So here's an example of the same kind of thing in a biomedical domain. So here's an item that our group has created about a gene called relin. You'll see it's all the same things here. It's got its unique identifier. In this case, we're talking about a different property. We're talking about the genomic start. There's the number of it. This is a number, okay, a number attribute. And we have for the qualifier, this is as of GenLoc assembly GRCH38. So you could have many different genomic starts depending on which assembly you're using. So that's important. And here is an expanded bit of references for that. So this information came from Ensemble Release 83, and it was retrieved on 19th January 2016. here's just one more example. So aside from basic property value pairs, like we were just talking about strings or numbers, the other thing you can do with Wikidata, and what makes it powerful, is that you can link one item to another item. So in this case, we have an entry about a gene, RELN, and the protein over here that it encodes. And that protein is another item in the database with its own attributes. That allows you to say effectively anything about anything. And so if you look at it as a database, what you see is a giant global graph. The statements link together into a queryable graph that you can access via query.wikidata.org, which is a Sparkle endpoint. Sparkle is a query language for the semantic web. And so coming now to what we're doing with it. Right now, over the past year or so, we've been actively seeding it with biomedical data. And so what we've done so far, we've added in items in Wikidata about all human, all mouse genes, and all of the Swiss prot, so the high-quality known proteins associated with each of those genes. We've also done a lot of that for microbial genomes, which I'll get to later. We've added all gene ontology terms to it. So now the entire gene ontology, including its graph relationships, are represented within Wikidata, so you can access it through that query system. We've also put in entries on all FDA-approved drugs. These were brought in from Drug Bank. And we have added entries in there for more than 9,000 human diseases. These are coming in through the disease ontology. And all of this is described in papers that have been recently accepted that I linked to before the talk. So what are we doing when we're saying seeding? Our seeds are basically, each seed item is a concept. And the main properties that we've added so far are really just links to different identifier systems. So if you look at an entry about a gene, you'll find about 8 different identifiers tied to it. If you look for an entry about a drug, you'll find it tied to 18 different identifier systems. And for diseases, about 11. And the reason we started with that is that what we really want to do is facilitate integration with other knowledge bases. We don't want to create, another independent silo here. The intention is to bring everything together into one place, and that really starts by making it possible to map from external sources into this system to unify all of them. It also makes it possible for them to be linked automatically via the semantic web, which we can talk about if anyone's interested. And so this is the goal. And what we want to do is produce a multi-community, what I call here a garden of biological knowledge. So, people at UCLA may add some connection, Scripps may add some connection, but no one individual group is going to add all the connections that we need to represent all the knowledge that we need to add. And so the goal is really to stimulate this kind of, very broad collaborative growth of this system. And so what I really want to convey to people is that Wikidata is not just about Wikipedia. It's really a platform for building biomedical applications. So this is what we've done so far. So far, we've taken a number of open data resources, we've mapped them into Wikidata, and we've used that to improve what we've been doing within the GeneWiki project in Wikipedias. One of the next steps there is working out good patterns for moving content generated by editors of Wikipedia and our content here back into Wikidata so that we can give feedback back out to the original data providers. As an example that happened automatically, we were importing data from the disease ontology, and people found that we were doing something wrong. So in that case, we had one disease that had several synonyms attached to it, just as synonyms, like they were the same, different words to describe the same thing. But in fact, someone in the crowd noticed that they were actually different diseases, and that information came back from that crowd to the authors of the disease ontology. They verified it, they altered their ontology, and it was improved by that process. So that's one small example of something we really want to enable with Wikidata. That's the improvement of the original source authoritative data sources by the crowd. But again, I'm going to come back to this. This is what makes it really exciting, because it's not just for Wikipedia. You can use this to drive any application you can imagine that would benefit from this data. You can build an iPhone application and sell it. You can build a widget for your biomedical website. Anything you can imagine. And I think that is where the real power of this and the really exciting point in its lifespan will start to happen, when people start doing that in a big way. So, as I said, the first application here is, in fact, Wikipedia. And so there's an entry about Relin on the right in the Wikidata viewer for it, where you can go and edit the content if you see fit. All of that is all driving the production of the info box displayed on the right of that page. So now every single bit of content that you see there is coming from Wikidata. And just to illustrate briefly why this is important to us, to our group, and to anyone else that has to work with Wikipedia, this is the change. So here's an example of what this looked like in Wikitext before we had Wikidata. This is exactly what it was in sort of version 1 of the GeneWiki project. So all of this information had to be represented. You see all the data is stored in there. So there's the gene ontology ID and there's the GenLo chromosome number and so forth. Now all of that data is properly stored in Wikidata as a database and represented in a way that we can put this one call to one script on any single human, any single gene, and it will produce that info box for us automatically. This is much easier to maintain than this. So this has been a huge win for us, and we're going to be rolling this out across all of the GeneWiki articles in the coming months. So the second application, as I said, I want to really emphasize that Wikidata is about enabling application buildings beyond Wikipedia. And so this is the second thing that we've built within the lab. Tim Putman within our group has worked with people from the Web Apollo genome browser development team to make it possible to show gene annotation data from Wikidata in the context of their browser. And so basically what happens, they launch the browser and they're able to use the query endpoint at Wikidata to get annotations that can be shown in it. And this very first prototype of this happened with I believe, B2K funded hackathon in San Diego in January, and they're working to make this a stable part of the Web Apollo code base. So that's a pretty exciting development for our team. So that brings me to the second part of the talk. We're gonna finish pretty early today, so we'll have plenty of time for questions if there are any. So I've talked mostly about what we've done with directly human relevant data, human genes, human proteins, maybe mouse proteins. But one of the biggest sources of community interest in this project is coming from microbial genetics community, the microbiome community. This information, much like all information, but it's like everything in bioinformatics, but it's more so. It's more widely distributed, it's larger, it's more difficult to query, and it's not structured at this point in any meaningful way. And I think because all of it is so new, the community is very well aware of all these problems, and so they're very interested in ways that they might be able to solve it. And so we've had people come to us from at least 3 now completely different institutes that want to use Wikidata to help solve this problem of integration of their content. And so, briefly, what we've done, as I mentioned, Tim Putman is a postdoc in our group who's tasked with loading Wikidata with microbial content and representing that knowledge. So far, he's added genes and proteins represented in a model that looks like this for 21 different genomes. He's on track to finish 120, I think, by summer. All of this, I think I didn't mention earlier, is done by code. Wikidata has very good APIs for working with it, for reading to it and writing to it. And so that work is steaming along. Also, a recently accepted paper out there describing it. So here's just a quick look at part of the data model that he has. So we have a gene here in blue. We can tell you it's found in the taxon. In this case, it's actually a strain of chlamydia, which is linked up to a whole long chain of parent taxons that will get you all the way up to bacteria. You see that that gene encodes a protein down here, which is its different unit. You see another gene over here from a different strain of E. coli, also encoding a similar protein linked together because they have the same EC number. So this is just a small fraction of the data model that he's come up with. In fact, he can represent things that are much more complex, and what he's really striving to do is make it possible to represent cross-species molecular interactions in the context of Wikidata, and he has examples in that paper of making that possible. So here's just a picture from that paper showing the complexity of what is possible to achieve even within this completely open system. And so, you know, we're very much at the beginning of this project, and I just want to say how much potential it has, and hopefully motivate other people to join us in making use of it. So I'm gonna wrap up early today and say that, you know, I raised two problems earlier with sort of knowledge bases in biology. First one being that they're not complete. The second one being that they need to be integrated. I hope I've given you some ideas about how Wikidata can help, and just to re-emphasize how I think this is going to work, the whole point here addressing both of those concerns is to centralize the content so that it's integrated in a way that's useful and allows you to walk across different data sources easily, while at the same time effectively distributing the labor of maintaining that knowledge base across a wide range of different people. And I think the most important thing here really comes down to, in the long run, the development of applications down here that serve very specific communities, such as the microbial genomics communities, that can let them feed their specific knowledge back into the central system where it will be integrated. We're at the very beginning of this project, but that, I think, is its key feature. And so I'm gonna wrap up there and just say thanks for the chance to talk to you guys, and thank you to a lot of people that are making this project come off. I'll start at the top with Andra Wagemeister, who was our initial developer on this project. He's been working with us for now, I think, a year and a half, and has done a lot of the work on the bots that populate Wikidata. Sebastian Bergstaller over here is a postdoc in our group who has also been principally responsible for architecting a lot of the code for talking to Wikidata, and is also the author of one of the papers I talked about. Here's Tim Putman, our microbial genomics Wikidata specialist. And over here is Elvira Matraka, who's a postdoc who's helping us work on diseases in the context of Wikipedia and Wikidata. Julia Turner has written all the code for showing that content within Wikipedia. Justin and Paul are helping us bring in the next phase of this project, which will be the links between genes and diseases. And of course, none of this would be possible without Andrew's support in getting us funding to do this and his guidance for the project. And lastly, I'll just mention Ginger Sung from our group, who adapted the Wikidata logo so it's more appropriate for biology and medicine. So I'm going to stop there, and we have plenty of time for any questions that there are from the audience. So thank you very much.
Thanks much. This is Susan. Maybe I could kick off our Q&A component. Can you hear me okay?
Yeah, please. Go ahead.
So as you were speaking, I was browsing through Wikidata just to get a sense and feel of how it would look for a user. And when people come in, so I looked at gene expression data, for example. When people come in with different experiments on the same gene, so the Wikidata will have each of those experimental data points added to it. Is that correct?
So two things I want to say about that. So let me bring up one slide just to come back to it. The structure here allows you to make multiple statements about that. So if you have different experiments that show the expression of a gene in different contexts, or are just in the same context, but run by different people, you can represent the results, all of them, basically. Okay. And so that when you make a query through the system, you need to be aware and make use of, the qualifier information that would, for example, tell you that this gene is being tested in this particular tissue or using this particular methodology, and the references about who did it, how they did it, and so forth. So from a knowledge representation standpoint, yes, all of that would be represented in there in a way that could be filtered at query time to get the data out that is most appropriate. But the other thing I want to really emphasize is that the Wikidata browsing interface that you see is really not what we want to show anyone. So that is there because you got to imagine that they have to create something that's completely generic. I mean, they have to be able to show data and let people edit data that is about Relin, this human gene, but it's also about operas in Sydney. There's no way you can make one application that can do a great job at that. And so that's why I'm really trying to beat on the concept here that we can make applications that are suitable for our specific audience using this data. So like the genome browser application, if that makes sense.
Yeah, that makes a lot of sense.
So my question goes a bit in the same direction. If there's half a dozen groups which all upload, let's say, interaction data, in Wikipedia, there's a fairly important role of the editors as moderators, as sorting out conflicts, etc. So what is the equivalent in Wikidata where somebody would at least discover, oh, in the upload from John Doe last week, they had actually an off by one error in their table and it's all rubbish. So is there something comparable like the Wikipedia editors?
Yes, absolutely, and that happens at multiple levels. So let me explain the process a little bit. First of all, it's not completely that easy to load a large data set into it. In order to do that, you'd have to do it programmatically via what they call bots, right? Now, if you're going to do a bot run, first of all, you have to have your bot account approved by the community. So not just anyone can go in there and put in 1 million statements without being verified by the community first. Secondly, the data structure that you use is determined by what properties you have access to. So in this case, I'm pointing to the genomic start property. And so there's a community process, and again, the Wikidata community has decided that you can't just add your own property as you see fit. And so you propose the property, say what you're going to do with it, and then the community can say, yeah, go ahead, that makes sense, and that gets approved and added into the system so that anyone can use it. And that's kind of one way that the data model itself is maintained by the community. And so given those initial constraints, once the bots are running, there are lots of other people's bots and people that watch the data coming in and do exactly error checking just like you just described. In fact, this is something that has hit our bots many times, that we've had people that, for example, last summer we had a big problem where we accidentally created a whole bunch of thousands of duplicate records. So multiple items for the same thing, which is obviously not what you want. Other people's bots detected that. They shut our bots down, sent us a message, and we were able to resolve it. And so just like Wikipedia, there's a large community of people that effectively patrol the data going in here. The key difference is that the patrolling is very much done, just like the data loading, by automated processes in Wikidata, because you can imagine there's two reasons. One, there's large volume. Okay, so that's kind of the only real way to keep on top of all of it, at least at the moment. And two, it's the stage of the project we're at. So for the moment, Wikidata is mostly something that is being used and edited by people that can code right now. That over time, if things work out, that will change, and then the volume of people coming in and manually eyeballing individual items will continue going up. I mean, in fact, Wikidata is actually the second most human-edited application within the MediaWiki universe already, and so that includes all of the Wikipedias, so Wikipedia Germany, Wikipedia, the only one that's edited and touched by human hands more than Wikidata is the English Wikipedia. And so the growth of that is really looking very good. And I think you could see if we do get past the threshold of the generation of applications that are specific for different audiences, you'll see that just explode. And so then you'll have many, many more eyes there to make checks on the quality of the data. Does that make sense, Henning?
Yes, thanks. That was a very exhaustive answer. I've got more questions, but I'll leave some space for others.
Okay. Sorry, I gave a short talk so I can give long answers to questions.
Okay, if there's nobody else, then I've just a detail question. The identifier on this current page, this is just random, or what is that? Q41 Forces.
So, yeah, so that is, you'll see it in the URL at the top. That is, sorry, but it's a new one. This is, so every item within Wikidata has its own unique identifier using that Q, or if it's a property, it's a P. So that allows them to give it their URL.
Okay.
And so we have contributed one more identifier into the identifier circus.
Okay.
So my apologies for that, but I think given the scope of it and the sort of generic nature of it, I think it couldn't be avoided.
Hi, can I ask a question to Salvatore? Can you hear me?
Yeah.
So it's more of, if you could elaborate a bit more on the microbiome data that you mentioned. The reason because I'm asking about that is that I'm somehow involved with the Human Microbiome project, and it is reaching like a phase where data are gonna be collected all together and be made available. And I see that you have a specific slide there on that. So I'm wondering if any of this relates to the HMP that was funded by the NIH, or there are it's kind of the data that are coming from all different places, or is there any plan to look into the HMP?
I don't know if we have a specific collaboration or intent to use the HMP data. So I can double check on that with Tim and Andrew, but my impression is that at this point, not specifically, but if it fits into the the general goal, then of course we would want to incorporate it.
Yeah, I think that if it does, for what I know, maybe wrong, it is not the largest microbiome data set that has been funded by the NIH. This has been put together in the first phase. Now it's in the second phase where more data will be generated. But yeah, it's just something to keep in mind if you proceed in trying to get all the microbial data in Wikidata.
Yeah, absolutely. And I think we're we're also working with Rob Knight's group at UCSD, who's definitely been a part of that, I assume. And there's even more data coming in fresh from them, and we're trying to figure that out as well. But I'll definitely make sure that we're on top of the HMP data.
Thank you.
Question on the data structure. So a lot of data, and here I emphasize data, in bioinformatics is, well, multidimensional. At least it's sort of metric style, like expression metrics, or it's multidimensional, let's say, when the time comes into very high dimensionality. And the Wikidata structure, to me, seems more focused on the derived knowledge. So the facts, like staying in this example, if you have an expression matrix, then I'm not quite sure how you would want to put the expression matrix in, but you would want to put in probably in spleen tissue, PNK1 is overexpressed. Is that a correct observation that it's more like it should actually be, at least in our domain, more be a wiki knowledge or wiki facts or wiki statements rather than wiki data?
Yeah, I think that's a very astute observation about what it is for. So I think you said it very clearly what it is best suited for, and there's definitely a limit to how much data and what kinds of data we want to and can put into it. So, for example, with the the microbiome data, a lot of that data, I mean, the great majority of it is in fact just piles of sequences, right? And that's not something that the actual primary sequence data is not something that's going to be put into Wikidata in any near future. What is going to be put into it is interpretations of it, just like you said, that this gene exists and is expressed at this level at this point in time. And then with the references and the qualifier information allowing you to go back and connect that out to an external primary source at NCBI or at HMP and so forth. And so, I mean, in that sense, it is very much more about representing sort of knowledge, whatever that means, versus primary data. And in terms of connection to primary data sources, what that means is that it could become a very useful index into them, if that makes sense.
Yeah, that makes sense. Thanks.
Any last questions?
Well, I've got one more.
Shoot.
I just don't want to be the only one. So you said that you are seeding this, or you opened the presentation with manual curation is not scalable. Mm-hmm. You have seeded the system, and you emphasized you seeded it with the good part of, for example, UniProt, namely the manually curated SwissProt, not the automatic part Tremble.
Yep.
So there's somehow, well, you can say, okay, Wikidata is just a better integration platform. There have been probably not 500 papers, but 5,000 papers about data integration in bioinformatics. Yeah. So one point might be it's just a better data integration platform. But can you comment a bit more on how you hope this will develop with respect to making a scalable knowledge management, basically what you referred to in the beginning of the presentation? How do you see the hundreds of thousands of individual scientists who ultimately generate the facts contribute? Because so far, I would say in bioinformatics, we have not been that successful in that area.
Yeah, sure. So, I mean, I'm going to come back again to the same drum that I've been beating here. I think what we need to do to make that really work, I mean, well, in principle, they could come to the Wikipedia page and press at the bottom of our new info box, there's an edit button, and they could go in there and edit it directly. So in principle, that could work. I mean, that could be a starting point. But I don't think that the hundreds of thousands of scientists that we're after here are going to do it that way. I don't think that that's the main way that we're going to solve the problem through Wikipedia. And that's why I really emphasize the need for these other applications to be created that are very much focused on specific domains, and these are showed in the context of people that are experts in those domains. I think to get to what we want to achieve, we want to be able to have people see editing interfaces and so forth where they don't even know or care that they're talking to Wikidata, right? They want to know that they're adding an entry to an item in their genome browser view that they have, and that behind the scenes it's getting integrated and used by everyone else. And that's how I think you achieve scale, by really distributing this access to the central repository and the editing ability in the context of applications that are suited to specific communities. There's another aspect to that. So I was just talking with Susie Lewis from the Gene Ontology and from the Web Apollo group about this. And so one of the things that she wanted to see was a way to track the edits made by an individual curator within their team on a genome. Because right now they have no good way of doing that in ways to sort of give them credit. And Wikidata could serve as a way to do this. So what you could do is say, okay, in the evidence statement that goes into Wikidata for a new piece of information coming from Web Apollo, for example, you could attribute it directly to the curator as an item in Wikidata so that they could, first of all, we could know who did it, and there's ways of doing that automatically, but we want to make it that explicit, and they could be credited for that. So what that leads towards is a way, like if you follow the nanopublication concept, of giving people credit for doing this kind of work. Because that's one of the main reasons that I mentioned earlier in the slide about the small number of curators, is that we don't get a lot of credit for this kind of work in science. And that's one of the reasons why it's a problem. And so Wikidata may play into this particular thing that BDGK is, I think, supporting, of giving people different metrics of success, different metrics of contribution that would encourage them to do things like edit these databases. So I think Wikidata could play into that. So.
Okay. Thank you.
Any other questions? All right. Thank you, Ben. That was a great presentation and great discussions.
Great.
That concludes today's conference. Thank you, everyone, for attending.
All right. Thank you. I'll see you soon, Henning.
Okay. See you. Susan, are you on the line?
I am on the line. Yes,
Did you
I'm
get
here.
my message in the message box?
Did you just send it?
Yeah, in the message box.
Oh, hang on. Let me. No, I don't have it. I'm sorry to say.
Okay, well, basically just, so are you, do you know already if you will come to the PI meeting?
I know that Salvador is definitely coming. I have approval to come, and now I have to very quickly rush to get the passport because I just didn't have that part done. So it's not very likely, but I'll try to push it through. But Salvador will be there, and so he will be able to talk about BD2K and the center and all the activities and represent us very well.
Okay. Let me know when you know for sure, but then I might have to, because we definitely need that part, so then I will put him in for that.
Absolutely. No, definitely put him on the agenda for at least 30 minutes, so he'll be there. And I'll try to come, and if I can't come in person, perhaps I could just phone in.
Yeah, that would be great. We'll definitely have a facility for that.
I'm still on the phone, so I heard you.